Hey everyone, I want to compare meiosis and mitosis today. So, first of all, do you remember mitosis back from chapter 3, I believe, in 141? It's been a while, so let's review this. So I have a cell here that has only one set of chromosomes. We have our maternal, I drew it in pink, and our paternal, I drew it in blue. Maternal from the mother, paternal from the father. Alright, so what do we do? So this is before mitosis, before cell division. We haven't replicated our DNA yet. So here we're in interphase, and here we've replicated. We're in prophase, and so you see we have now two maternals, two paternals. In metaphase, the chromosomes line up at the metaphase plate, and then they separate. We end up with two genetically identical cells, right? They look exactly like the parent cell. Do you guys remember that? Mitosis. And where does that happen? I like to say mitosis happens in my toes. It really happens all over my body. But meiosis happens in my ovaries. Like oosis, ovaries. Mitosis happens in our toes and our other non-gamete cells. All right? Now, how about in meiosis? What's the goal of meiosis? Well, when we have a sperm and an egg come together, we can't have them both have 46 chromosomes. They have to have one. They both each have to have 23 chromosomes. And so what we need to do is we need to go from 46 down to 23. And we want to do that in such a way that we have the greatest genetic variability. We need to have all the genes. We need every gene present, but we need to make it as unique as possible. All right, so here we are, interphase, prophase. We're going to copy the DNA. Well, my goodness, these look pretty similar. All right, but then here's where it starts to really change. In meiosis, what's going to happen is these have the same genes, right? Because I only have one chromosome in this picture. That's all I'm able to successfully draw. But if this were humans, we would have 23 sets of these. So 23 blue X's, 23 pink X's. And what's going to happen is these are going to come together, like you see here, and they're going to form a tetrad. Let me draw a tetrad here real quick. So maternal, the replicated, and paternal. And let's go over a couple definitions here. So these are sister chromatids, right here and right here. This is a tetrad. They're going to be really kind of joined by here, and that's going to be synapsis. Tetrads joined by synapsis. So you have sister chromatids. It's a replicated chromosome, all right? And so you have the chromosome and its copy, which is its sister. Chromosome, copy, sister chromatids. So you have two sets of sister chromatids coming together to make a tetrad. Think tetra being 4, and there are 4 chromosomes. Now, how about the genes on these? They have the same genes, but different alleles, different copies of the genes. Although really, these two are identical, and these two are identical. So maternal, paternal, but they have the same genes, not necessarily the same alleles, maybe different. So one might have blue eyes, one might have brown eyes, but there's still the gene for eye color, if that makes sense. One might be tall, one might be short. One might have a tall gene, one might have a short gene, but it would be the same gene for height, just a different copy of it or allele. which we'll get into next chapter. All right, so we have the tetrads. They've come together by synapsis. Now look at this, crossing over. What is that? Because remember, look at how they're lined up here. They are lined up single file, basically. Here we're going to line up as a double line. And it's this coming together here that's going to allow them to do crossing over. What is crossing over? Well, it's these chromosomes, they're kind of next to each other, and they have the same genes, so they can swap. And this is a great thing because it increases our genetic variability. All right, so I've kind of drawn it in. You can see how they've swapped genes here and swapped genes. Because they have the same genes, they can swap. So they can say, you know what? I want your eye color. Oh, I'll take yours. It's a little bit like if you have a card game, and you've got a bunch of cards, and let's say have you ever played bridge? I think you can do swapping in bridge. I'm not really good at cards. Or it would be like a long time ago in elementary school, we were able to swap lunches. Be like, oh, I don't like what my mom packed me. I'll trade you my sandwich for your orange. Something like that. And it's similar. They swap genes, and really increases the genetic variability because now these are going to be pulled apart, and you're going to have four genetically different daughter cells. But before we do that, let's take a look at how they're going to be pulled apart. So, got your spindle. All right, how are they going to pull apart? Because they're not lined up single file, they're lined up double. So they're going to get pulled apart. And you know what's really interesting? Even though if you were to take the DNA out and weigh it, you have the same by weight as you do over here in the diploid mitosis daughter cells. But after this first division, you're going to be haploid. And why is that? Well, look at this. Does this one have the mother and the father? It only has a little bit of the paternal genes. And that's only because of crossing over. How about this one? This one has almost exclusively the paternal. It only has a little bit of maternal, and that's because of crossing over. I love how the book gives an example of this. The book talks about how if you were to order your lab book and your lecture book, but what if they only sent you 2 lab books? You would be saying, wait a second, I only have half the books I need. And the bookstore might say, no, you have 2 books. You'd be like, no, but it's only half of what I need. Well, here, they have 2 chromosomes, but they're haploid because they don't have one of each. It's 2 paternal or 2 maternal. They don't have the full double set. Does that make sense? So they both have 2 chromosomes, just as if you and a classmate, maybe one would have 2 textbooks and one would have 2 lab books. By weight, you would have 2 books, right? But they wouldn't be the 2 different books that you need, if that makes sense. So after the first division in meiosis, we're haploid, all right? That one's a little bit hard to understand, I think. So let me know if that's confusing. It's important and it's challenging. So after the first division, you're haploid. It's like having 2 lab books or 2 lecture books instead of lab and lecture. They both have all the chapters, just like they have all the genes, but it's not the 2 different versions that a non-gamete cell would need. So here we're haploid. All right, now we have another division. All right. Draw our spindles. Pulls it apart, and what do we get? 4 genetically unique daughter cells. So in meiosis, we undergo 2 divisions, but we're haploid after the first division. We have tetrads, we have the crossing over, we have the double line at the metaphase plate. Right? In meiosis, they line up single file. The sister chromatids, I should say, line up single file. All right, and we only have one division, and we are diploid. We have both the mother and the father. Here, I have father, I have mostly father, a little bit of mother genes crossed over, maternal. Here, mostly maternal, a little bit of paternal genes from crossing over, and here, only maternal. Now, can you imagine the variation that you would get if you have 23 of these? Because can you imagine how if you have 23, well, maybe this is a maternal, but you could have a paternal, because they line up independently, independent assortment. So it could be, your genetic variation is just remarkable when you get to 23 chromosomes. And that's a wonderful thing. We need all the variability that we can get. Alright, so meiosis versus mitosis, 4 daughter cells, 2 daughter cells. Haploid cells, diploid cells. 2 divisions, but haploid after the first. 1 division, diploid. Lining up double file, double line at the metaphase plate, forming those tetrads, right? And single file of sister chromatids. And we have crossing over in meiosis. Happens in our gametes, happens all over the rest of our body. All right, does that make sense? Sperm cells, egg cells. The only time we need that. All right, everyone, let me know if there was anything confusing here. It's an important concept.